Hello, and thank you for coming to check out my poster. My name is William Myers. I'm an undergraduate senior at Rutgers Camden, currently applying for PhD programs, and I will be talking to you today about the work I'm doing remotely with the Fredolino Lab at the University of Michigan. So, we're all probably familiar with the idea of bacteria being pathogens, and they are also used as research and industrial tools. But whatever the context, optimal performance of bacteria is dependent upon their phenotype, which for bacteria is mostly a result of how they regulate transcription of their genome. In addition to the more familiar example of transcription factor proteins, bacteria also utilize nucleoid associated proteins to regulate gene expression. Interestingly, when you take a global view of protein bound DNA, you start to see these long regions of the bacterial genome that are packed with these nucleoid associated proteins. These regions are referred to as extended protein occupancy domains, or EPODs. More specifically, we define EPODs here as genomic regions a kilobase or longer that have very high and sustained protein occupancy. Typically, EPODs silence the genes that they cover. The broad hypothesis of this work is that we can investigate the presence of EPODs to inform our understanding of how bacteria regulate their genome in response to conditions of interest. Specifically, my work here looks at EPODs in two contexts. First, we're going to talk about DNA methylation in E. coli. So in E. coli, DNA methylation is involved in repairing DNA replication errors, chromosomal replication, and transcriptional regulation. The two methyltransferases that do the majority of DNA methylation in E. coli are known as DAM and DCM. The top panel in figure 1 is an example of how protein occupancy blocks methylation of DAM target sites in the promoter region of an operon that's associated with virulence in pathogenic E. coli. Now, this is a local example of how protein occupancy inhibits or allows DNA methylation, but what if this kind of crosstalk between protein occupancy and DNA methylation happens across the whole genome? And is it only that protein occupancy informs DNA methylation, or can DNA methylation also inhibit or enable protein occupancy? To answer this question, the Fredolino lab grew wild-type E. coli in addition to deletion mutants of DAM, DCM, and a double deletion mutants of DAM and DCM. We first attempted to answer the question of if the different methylation states of these strains would impact protein occupancy at methylation sites. The results of this are shown in the left panel of Figure 3, and you can see here that there are very few discernible differences between the strains. And we continue to see very little difference between occupancy at these methylation sites, even when we look specifically at the representation of these methylation sites in EPODs, which is shown in the right panel of Figure 3. So ultimately, from this investigation, we can pretty soundly conclude that the different methylation states present in these strains is not significantly impacting the protein occupancy across all of the methylation sites in the E. coli genome. Okay, now we're going to talk about biofilm formation in Vibrio cholera. So at least two of the factors that lead to biofilm formation in V. cholera are increasing local cell density and high intracellular levels of the second messenger cyclic di-GMP. And so to investigate the role of EPODs in this context, the Fredolino lab grew wild-type V. cholera in addition to a deletion mutant of a transcription factor regulated by cyclic di-GMP. They then took samples over 4 different time points after exposing their cell cultures to one of 2 different plasmids. A schematic of this experiment can be seen in the right panel of Figure 4. One plasmid contained an inducer that would greatly increase cyclic di-GMP levels. The other plasmid was a control to look at the effects of increasing cell density. I then found how large-scale protein occupancy of the genome changed over these time points. In Figure 5, we're looking at the protein occupancy at the genomic region of 2 neighboring virulence-associated operons. And we can see that in particular there are drastic occupancy differences at the 15-minute time point after introducing the increasing cell density control plasmid. And while this is a local view of occupancy changes, we can see by the heat maps in Figure 6 that globally, across at least the first chromosome of V. cholera, that there is a sizable difference in the location of EPODs between the plasmid conditions at specifically the 15-minute time point. And this difference is really muted when we look at the transcription factor deletion mutant. Overall, we can see from these two examples that looking at large-scale protein occupancy, and specifically EPODs, we can start to form really interesting questions about transcription regulation in bacteria responding to conditions of interest.